And the topic for today's lesson is enzymes. So without wasting time, let's go to what we have today. First of all, here we say enzymes are biological catalysts. We already know the meaning of catalysts from chemistry. We say catalysts are substances that can speed up the rate of chemical reaction. They speed up the rate of chemical reaction, but they're not used up in the reaction. They don't get used up. Okay, they don't lead to the formation of the product. They only aid to speed up the reaction. So that's what catalysts are. Now we say enzymes are biological one. Remember, catalysts. They are substances that can speed up the reaction rate. Now we say the enzymes are biological catalysts in this regard. Okay, so here we're going to look at the functions of enzymes or the functions of biological catalysts, and we're also going to look at how they be classified and give examples of enzymes. Okay, so we say function. One of the things enzymes do is that they digest large food molecules into small ones. They also help to break down food substances in the cell to increase the release of energy in the cell. They do so many jobs, okay? And then we look at their example, okay? So we have things like carbohydrates, enzymes like carbohydrates, which are responsible for carbohydrate molecules or carbohydrate food. We have enzymes like protease, which are responsible for protein food. We have enzymes like lipase, which are responsible for lipid or fat and oil. Now let's start with the first one, which is a catalyst. Like we said before, a catalyst is what? Is a substance that increases the rate. This word must be there. A substance that can increase the rate. The rate will be the rate of chemical reaction. Rate of what? Rate of chemical reaction. Without being chemically used up by the reaction. Without being used up. It's not going to be part of the things to form the product. You know, we have substance, or what we call the substrate, and being acted upon by the enzyme when it fits into an active site of an enzyme, which we're going to talk about, and then producing the product. But catalyst itself, something we call catalyst, they don't get used up. They don't get broken down. They don't get digested also. They only help to speed up the rate of reaction. Now, what are enzymes? If we know what catalysts are, we say, first of all, before we talk about enzymes, we say manganese dioxide can be used to speed up the rate of the breakdown of hydrogen peroxide. So when you want to break down hydrogen peroxide, you know hydrogen peroxide is H2O2. It's H2O2. This is what we call hydrogen peroxide. So if you want to break it down into water and oxygen, if you want to break it down into water is H2O. If you want to break this hydrogen peroxide into water and oxygen, we use what we call manganese dioxide as an enzyme to speed up this reaction, to speed this chemical decomposition of hydrogen peroxide into water and oxygen. So such substance that will not get itself used up, but it will participate or speed up the rate at which hydrogen peroxide will decompose into water and oxygen is called a catalyst. It's called a catalyst. So like this black one that we add to this, the white one is the hydrogen peroxide, okay, in this bowl. The white substance is the hydrogen peroxide in this bowl. And then this black we add is called the manganese dioxide. This manganese dioxide is a catalyst. It's only speeding up the rate at which this will change to water. It will speed up the reaction. This one will still remain. Okay, so that's what we call a catalyst. Now let's go to enzymes. Enzyme itself is a protein. That's why we always encourage you to eat protein, so that you have enough enzymes to digest your food. Enzymes are biological catalysts. You need to know, you need to be able to say this even in the exam. Enzymes are biological catalysts. Then, before you now follow up with what the meaning of a catalyst is, you say they ask you what are enzymes. Enzymes are biological catalysts that can increase the rate of chemical reaction without being used up in the reaction, without itself being changed. So enzymes are biological catalysts. That's the only difference. This one, catalyst could be physical substance. But enzymes are not physical, they are biological substances. So enzymes are they are catalysts, but they are biological type. They are biological catalysts, which helps to speed up the rate of chemical reaction without them themselves being used up in the reaction. So in simple terms, enzymes are protease. Enzymes are protease. So what are enzymes? If you give these two, you get the 2 marks. Enzymes are biological catalysts that help to increase the rate of chemical reaction without themselves being used up. And then you say enzymes are proteins. So if it's 3 marks or whatever marks, you're going to get that marks. Now let's go to the next thing. So what reactions do enzymes catalyze? What reactions do enzymes catalyze? What reaction do they act upon? Well, okay, when we say do enzymes catalyze, we ask you which reaction do enzymes act upon. Number 1 type of reaction they act upon is reaction in digestion. During the digestion of food, enzymes play a very active role in the breakdown of the food. You know, digestion is simply the breakdown of large food molecules to smaller food molecules. So during the breakdown of food, enzymes does the job. They help to do that. Food is broken down by enzymes during digestion. That's the first thing, right? During the large food molecules like proteins are broken down. When they pass through enzymes will help to break them down into a smaller unit called the amino acid. Okay, fat or lipid will be broken down into glycerol and fatty acids. Carbohydrates will be broken down into maybe maltose to glucose, and so on and so forth. Now let's take a look at example of digestive enzymes and the work they do. Let's take a look at the example of digestive enzymes. So you need to know all this in the exam. You need to give the enzyme and what does it digest. Amylase. You will hear of amylase. Amylase break down starch and change it to maltose. Amylase breaks down the starch, they digest starch and reduce it to what we call the maltose. Okay, you can remember this with malt. Those who drink maltina, malt from Guinness or from this thing. So amylase is an enzyme that breaks down starch. Starch is irregular cellulose or starch, irregular carbohydrates we've studied. The polysaccharides. They are the polysaccharides. In chapter 4, we take a look at polysaccharides, monosaccharides, disaccharides. Okay, starch are regular polysaccharides. They are large, giant macromolecule. Okay, they need to be broken down in the process of digestion. So the first stage, digestion is in different stages. So the amylase break it down, not straight to glucose. They break it down first to what we call maltose. That's the job of amylase. It is not the job of maltase. There's another enzyme they call maltase. Okay, another enzyme called maltase that will now break the maltose into glucose. So the job of maltase is to, so you remember this by saying, okay, maltase will break maltose to glucose. Maltase will break what? Maltose to glucose. It is the one that have AES, that is enzyme, that end with AES. Okay, is a name. The ending of enzyme is AES. 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 Take a note of that. Take note of that. So I will still explain how enzymes are named, how they are named, but they end with AES. So the end, the AES is the enzyme. So in the enzyme, so you don't get confused. You see maltose, say, oh, is it the maltose that is the enzyme or the digestive food that is enzyme? So maltose will confuse you with maltase. Remember how you know what is enzyme is that enzyme ends with AES. Amylase, maltase, protease, lipase, all of these, they end with AES. So that's the characteristics of enzymes, the naming of enzymes. We first derive the first part of the name from the food they digest, like protein. You see, prot is from protein. This protein is from protein. And then the AES is the enzymic naming, the enzymic naming. So that's how you know enzyme. Lipid, fatty acid, they're called lipid. The LIP is from the lipid. Then the AES is the name. So this is how we know that. We talk about them soonest. So we say maltase is the enzyme that is responsible to digest maltose to glucose. Now protease, although protease can be divided into two, pepsin and trypsin, which you're going to study in the digestive system in chapter 6, I guess. Okay, we're going to study that in chapter 6. Proteins, either chapter 6 or chapter 7. Proteins are divided into Pepsi and Trypsin. The Pepsis are in the stomach, the Trypsin are in the small intestine. Okay, we'll talk about that later on. But for now, just know that the proteins is the enzyme that digests protein food and break protein food into the smallest unit of protein called the amino acid. They digest protein to what we call the amino acid. Then the lipids digest fat to fatty acid and glycerol. You know, fat food or fat and oil, they broke it down into fatty acid and glycerol. We studied this in chapter 3, in chapter 4, when we were doing something like biological molecules. Okay, chapter 4. Okay, so that's the enzymes and the indigestion, the kinds of enzymes you find in digestion. Now, what reaction do enzymes catalyze? What type of reaction do they catalyze? One of the reactions they catalyze is the digestion of food, right? They help in the reaction in the digestion of food. One is the reaction in digestion, they help in the digestion. How do they do that? Now, let's take a starch molecule. This is a starch molecule. You know, the polysaccharides or the proteins or the lipids, they are giant macromolecules, okay? Now, the job of the enzyme is to break it down into small units. This bit. Break it down into one piece, not linked with chain, okay? That's the job of the enzyme. So, action of the enzymes, such as the amylase, you know, amylase, we break down starch into what? Into maltose. So, amylase and maltase, you know, maltase is also another enzyme. Amylase and maltase will eventually help to break it down into what? Glucose. First, amylase will break it down to maltase, maybe two units. Maltase is like maybe you are breaking it down into two units. That's the meaning of maltase. Then to break it down into one unit, is the help of maltase enzyme to make it glucose, which is now one unit, like this single unit. So the single unit, why do we have to break it to single unit? Only single unit can diffuse or with the help of active transport can move through this cell membrane. You know, this is cell membrane. These are pores. In between cell membrane, we have pores. We have what we call the pores. Okay? So only this smaller unit can actually move through the pores and enter the cell, inside the cell. Can enter inside that cell, digest the area where the food is needed, the part of the cells of the body where the food is needed. So this chain may hang here. If you don't break down the food, the chain may hang, may not be able to pass through here. So we need to break it down to small units so that each single unit can pass through either through the process, whatever process, which we study in chapter 3. The diffusion, osmosis, and active transport, to pass through and enter the cells as required. Now this for carbohydrate or starch. Now this is protein. Protein have different single units. Okay, have different. Some are circular, some are robust, some are rectangles, some are triangles, some are square, and so on and so forth. Linked together with a chain. That's what we call a protein. So protein contains so many singular different units. Now, when the enzyme called proteins act on it, the actions of the enzyme called proteins, to break it down into each individual unit, to break it down, and each individual unit is able to diffuse or is able to, with the help of active transport, able to move into the cell of the body. So that's what is taking place. Now the second action of enzyme, or the second reaction enzyme undergo, is what we call the reaction that break up or break down complex substances. Reactions, enzymes take part in the reaction in the body that helps to break up or break down complex substances. Now let's take a look at build up, not break up. Build up and break down. Build up is to synthesize. Break down is to decompose or reduce to smaller units. Now the build up complex reaction that enzyme undergo, one of it is amino acid can join together to form protein in the body. You know, when the amino acid reach the body cells, they can synthesize, they can join together, they can link together with the help of enzymes. Enzymes can help to link them together to form protein. That's build up. Build up means you can again join the smaller unit to form the micromolecule. to form the macromolecule called the protein. So each tiny amino acid can be linked together with the help of enzymes, to form what? To form the protein, to form the protein. Okay, so that's the build-up. They can join together to form the protein. That's the build-up complex reaction. Now the breakdown complex reaction is that we say cells break down complex substances to simple substances. Okay, for example, like the hydrogen peroxide we talked about. Hydrogen peroxide can decompose with the help of a catalyst and enzymes to break it down to water and oxygen. Okay, so it is enzymes that break this complex one to what? To smaller units. This one is already clear. We explained this earlier on. It's only the build-up we didn't talk about. So the build-up is smaller unit can join together to form larger one. Enzyme catalyze practically all the chemical reactions that occur in our body. All the chemical reactions, all the metabolic reaction that is taking place in our body, it is enzyme that will catalyze them. Either those that will break down to smaller one, enzyme will coordinate it, will help to break them down. Those that will synthesize or build up, enzyme will also help to carry those ones out. Okay? Depending what the body needs, enzyme helps in. Now, how are enzymes named? How do we name them? We talked about this. We said the name of each enzyme shows the substance on which the enzyme is acting on. For example, they all end in AES. Enzymes, their name end with ASE. Like I've explained earlier on. They end with what? ASE, as we have explained. But the name of the enzyme comes from what is acting on. For example, the enzyme called lipase is coming from what? Lipid. Fat and oil, they are called lipid. They are called what? Lipid. Fat and oil, they are called what? Lipid. Collectively called lipid. Okay? So the lipase, which is an enzyme that acts on fat and oil, the lip is coming from the lipid. This lip is coming from the lipid, and we add the end of the name of enzymes. All enzymes, we end them up with AES. So you add AES to it. That's why it becomes lipase. That's how the name is named. Okay? This is the substance. The lip is the substance the enzyme work on. Enzyme is working on the lipid. Since it's working on lipid, the name comes lip. Then the AES is the end of the enzyme's name. So that's how we got that name. Now let's move on. So now we know how we name enzymes. Now we go to the next one, the second section. How does an enzyme work? How do they work? Remember, we say enzymes are biological catalysts. They can form an enzyme substrate complex. So how they work, the shape of the enzyme, the shape of the food that they want to act on, and so on and so forth, the active site. These are the areas we want to talk about now. Enzymes substrate complex. So they form enzyme substrate complex with a substrate that can complement the 3D shapes of the enzymes. Enzymes have what we call the 3D shape. They have a 3D shape, and the food that they want to break down, or the substance they want to break down, are called the substrate. They are called the substrate. Now this substrate, the shape of the substrate must fit into the active site of the enzyme. Must fit into the active site of the enzyme. In the same way, a key will fit into a lock. In the same way, a key will fit into a lock before the key can open the lock. Okay? In the same way, a key will fit into a lock before it can open the lock. And when that happens, they break down to product called final unit called the product. Okay? When enzyme act on the substrate, they produce, when they fit, they produce what we call the product. So in the exam, I think in one time we answered the question from enzyme in the past papers. Okay, so you will see the active site, and then you see the enzyme and the substrate if they fit. If they don't fit, then such enzyme cannot catalyze such food. Now how does an enzyme work? We say the substance which the enzyme wants to act on is called the substrate, which I have explained. The food substance, like this one, is called if this is, for example, this is an enzyme anyway. This is an enzyme. This is a practical scenario of an enzyme. Take it with a 3D shape. A 3D shape of an enzyme. This place where the lock, the key we enter, the key is called the substrate. And this place where the substrate we enter, we fit in, is called the active site. It's called the active site of the enzyme. Now, each enzyme has a groove on its surface called the active site. A groove, like a hollow hole where the food will go in, the food that he wants to break down. Now, this groove is called the groove on this protein is called the active site. Is that okay? Now, only a substrate with a complementary shape that can fit into this active site will go into this place. Only a substrate with complementary shape that can fit into this active site will go in here. Just like we explained that it's like a kind of lock and key scenario. Lock and key. The substrate is the key. The food you're bringing is the key that we open this lock. The enzyme itself is the lock. Where you go into is the active site. Okay? Now, when the substrate binds to the active site of the enzyme, we have what we call, when it fits in, we call it the enzyme substrate complex. We call it enzyme substrate complex. This is also a question in the exam. The enzyme substrate complex. Okay, when the substrate fits into the active site, that process is called the enzyme substrate complex. Now let's explain them looking at a substrate. This is a substrate. This is an enzyme. An enzyme is a 3D shape. Okay? This is a substrate. A substrate is just a food substance. It could be a protein food, a carbohydrate food that wants to be digested. Different enzymes have different shape, different active site shape. So it's only food that fits in. If you take a protease, it's only a protein food that can fit into a protease. That's the meaning. That's what we're trying to say. A carbohydrate food, the shape will not fit into a protein enzyme, the way protein enzymes are designed. So they will not fit in. Only a protein food can fit into a protease enzyme. Is that okay? And it's only a starch food that can fit into amylase and maltase. Amylase and maltase. Only a starchy food that can fit into amylase and maltase. So that's what we mean by saying that only the food substance or the substrate that can fit into the active site of enzyme goes it to form enzyme substrate complex. Now, enzyme has a specific 3D shape and it has a depression. We can call it a groove or a depression called the active site. Depression means something that goes in. Okay? A depression or a groove that is called the active site. Now, the substance on which the enzyme acts is called the substrate, like we have explained, right? So this is the first scenario. This scenario is showing what? It's showing a substrate entry and active site. This scenario is showing a substrate that is entry. You can see the arrow. This is the substrate and its entry and active site. Is that okay? Then, the second case, only the substrate with a 3D shape, only the substrate with a 3D shape. So if the substrate is not a 3D shape, obviously cannot fit into a 3D shape enzymes. Only the substrate with a 3D shape, complementary to that of the active site, that is complementary to this, that can fit into this, that's the meaning of complementary, that can fit into this, will go in here, right? We go in here. Now the enzyme is just like a lock and key scenario like I have explained. The key is the substrate. This is the lock. And this is where the key going into to open the lock. It's just like a lock and key hypothesis. Is that okay? A lock and key hypothesis. And this might be in the exam. Enzyme substrate complex is modeled as what? Lock and key. Or a lock and key. Which of the following hypotheses is used to model the enzyme substrate complex? The hypothesis is called the lock and key hypothesis. It's called the lock and key hypothesis. Lock and key hypothesis. Now this results in the formation of what? Enzyme substrate complex. Now, This second diagram is showing the substrate binds into an active site of the enzyme. The substrate binds into the active site of the enzyme. Now, the third scenario, while the substrate is attached to the active site, a chemical reaction occurs. Chemical reaction will take place. That's the third scenario. Once the substrate is attached here, you can see some broken line here. Some broken lines are shown here. So it's showing that a chemical reaction is about to occur or has started occurring. Now the substrate is converted to product, meaning it's broken down into two. You can see here. Once this comes, some chemical reactions start to take place. So chemical reaction occur while the substrate is attached to the enzymes. So this is the third scenario, and the final scenario is that it will break this substrate into two, and the two will now start to leave the product. These two are called the product now. They are no longer called the substrate. The product leaving the active site. Once it's broken down, they can no longer fit in here. They have to leave. So it means digestion has taken place. It means this enzyme has helped to break down the food or speed up the breakdown of the food into smaller units that can diffuse or that can, with the help of active transport, enter the cells that is needed through the pores membrane. Okay, so that's that for section 2. Now we're going to section 3. And section 3 is the characteristics of the enzymes. We already said enzymes are biological catalysts that can do so many things. Let's look at the things it can do. The first one is, what are the characteristics of enzymes? The first one is that enzyme can catalyze reversible reaction. Enzymes can catalyze reversible reaction, meaning reaction that can go both sides. Okay, enzyme can catalyze them. Any reaction that can, whether they are isothermic or what type of reaction, enzyme can catalyze them. Okay, so enzyme can catalyze or speed up the breakdown of reversible reaction. Okay, that's the first one. For example, a good example is carbon dioxide is a gas, react with water, they form what? Carbonic acid. Carbon dioxide react with water will form carbonic acid. But this will not happen if there is no anhydrase. Carbonic anhydrase is the enzyme that will speed up the process to make this acid. Is that okay? So that's one job they do. Enzyme catalyze in case they ask you the functions of enzyme or characteristics of enzymes, you say they can catalyze reversible reaction. Number 2 is that enzymes are catalysts. Now from the definition, you take it as a function or you take it as a characteristic. Enzymes speed up the rate of chemical reaction that occur in cell. They speed up the rate of chemical reaction that occur in cell. Enzymes, they help to speed up any chemical reaction that is taking place in a cell. Number 3, every enzyme has an optimal pH at which it is most active. Optimum pH. Now, here you need to know in the exam, pH, one of the factors affecting the rate of reactions of enzymes is pH, and another one is temperature, and another one is concentration. So there are 3 very notable factors you need to take note in Igcse. One is pH. Now, enzyme, which pH do enzyme at best? pH between 5 and 9. But they are optimum in 7, pH 7. Okay, pH from acid, basis, and salt. We've studied pH of this acid, basis, and salt. So from 1 to 6 is acid. From 8 to 14 is alkaline. 7 is water, right? So enzyme at best in a pH between what? When you are in 5, it's not too acidic. 1 is very acidic. 2, 3 are very acidic. 5 is less of acidic, it's diluted acid, it's not so acidic. And so meaning enzymes do not add super best in super acidic solution or super acidic soil. Enzymes don't add or super acidic stomach. There's level of hydrochloric acid in our stomach. So enzyme, if you want enzyme to act very well, not to acidic environment. Okay? The best of it is a neutral environment. The maximum, when they ask you for the optimum pH of an enzyme, optimum pH is what? 7. The optimum pH is what? 7. This is the optimum. That's the best one for enzyme to act is water solution. But it doesn't mean it will not act between 5 and 9. 5, it's increasing the rate of reaction. But it does best. It reaches peak at pH 7 and it declines 9. So between 5 and 9, this is the pH with which enzyme acts in our body. So at pH 4 or above 9, 4 means you are now this way. Enzyme will not act well. Enzyme will denature. That's what we call denature. So at pH 4 or when you are above 9 or when you are below 5, like 4, 3, 2, 1. So all those pH below 5 or above 9, when you are now above like 9, 10, 11, 12, 13, 14, when the alkalinity is increasing as you go this way, acidity is increasing as you go this way. So those pH, when you are too alkaline or too acidic, the enzyme will what? Denature. What do we mean by denature? It will disfigure the active site of the enzyme. That natural 3D shape of the enzyme will denature, will disfigure. We no longer, we lose its structure and complexity. So the active site of the enzyme loses its original shape. To lose its original shape. That's the meaning of being denatured. So when you say, when you increase the acidity to 1 or alkalinity to 14, then the enzyme will denature. The enzyme will lose figure, will disfigure. So the substrate will not be able to fit in to form enzyme substrate complex. The substrate will not be able to fit in order to form what we call the enzyme substrate complex. So as a result of that, the enzyme will denature and you will not be able to digest the food or break the food down to smaller unit. Okay, so still on the characteristics number 3 or characteristics of the enzymes. Number 4 now, we're in number 4. Enzymes are required in minute amounts. That means small amount. You don't need so much enzyme to break so much large food. You just need small amount of enzymes to catalyze a very large number of chemical reaction. So we don't need so much, we just need little. Okay, that's why we are safe, even as we eat less of protein most of the time. You just need small of it to help you catalyze the food. Number 5, enzymes are specific in action. This is very important. What do we mean by specificity? It means that only any carbohydrates, which include amylase and maltase, we digest carbohydrate food. You cannot bring a amylase to go and digest a protein food. They are very specific. They're very specific. It doesn't mean that because enzyme digests food, any enzyme in your body will digest any food in your body. No. Enzymes are very specific in their action. Only a substrate with a 3D shape that fits into the active site of the enzyme to form that enzyme substrate complex, we undergo into that enzymic reaction. We undergo into that enzymic reaction. So only a substrate that fits in. A carbohydrate food will not fit into a protein active site. Will not fit into a protein active site. That's the meaning. In summary, that's what we're trying to say. So it's only a substrate with a shape complementary to that of the active site, right? That can fit into the enzymes to form the enzyme substrate complex. So that's what we mean by enzymes are very specific in their characterization. Number 6 is that every enzyme has an optimal temperature at which it works best. Just so we did for pH, enzymes work best in optimal temperature. And I think the optimal temperature is around 37. Okay? Here will be around 40, 41 or something like that. So, but the optimal, where the enzyme work best, is at C. If they ask me the enzyme, which of the following temperature, they will give it in figure, like in a diagram like this, figure. They maybe here is 28, here is 30, here is 37, here is 38 or 39, here is 42. So which one is the optimal temperature for enzymes to work best is 37, somewhere here. So you should be able to look at the rate of chemical reaction when it's plotted against temperature. This is how the graph looks in the exam. Okay? So we say increasing the temperature from low to optimal increases the rate at which enzyme will act. From low temperature to optimal temperature, the enzyme will, the rate of chemical reaction, the rate at which the enzyme will break down the food will be faster. But when you go beyond the optimal temperature, it will slow down the rate at which it will act. Increasing the temperature above the optimal temperature causes the rapid decrease in the rate of reaction. Rapid decrease in what? You see the graph going down, means it's a decrease in the rate of the reaction. This one, the graph is going up and it reaches maximum at optimal temperature. Okay? Increasing temperature increases the kinetic motions of molecule usually. Because of that, because the kinetic motions of molecule increases, it will result to an increase in the rate of effective collisions of the molecules of that food substance. Effective collision of the molecules of the food substance and the enzyme. The food substance is called substrate, don't forget that. So when that happens, it will produce the product at a faster rate. And that is what we call excuse me. That's what we call the optimal temperature. So please remember to note the graph as it comes in the exam. Is that okay? Now, what causes enzymes to denature? What causes them to denature? When you go beyond, first of all, what is the meaning of denaturation? We call denature. What is the denaturation? Is the change in the 3D shape. Now, this is the enzyme. This is the active site. This is the substrate that fits into this enzyme. So when you increase the pH, when you make it too acidic or you make it too alkaline, it can disfigure this shape. This shape that is like this can be disfigured like this. So when the substrate comes, substrate molecules come, no place to fit in. So it can no longer break this food. The enzyme can no longer break this food because it is denatured. Okay? So, but if it's not denatured, the substrate molecules can fit into the active site and then break down this food into products. Break this giant macromolecule into a smaller unit called the product. One of the factors is too much acidic environment or too much alkaline environment for the enzyme. Number two is increase in temperature above 45. When you make the temperature to be above 45, you can denature the temperature of the reaction temperature of the environment. If it's above 45, can denature the enzymes. Okay, can denature the enzyme. Enzyme don't require too much high temperature. You just need around 37, just like the normal body temperature. Normal body temperature is around 37 degrees Celsius. So that's what the enzymes also need in order to give you optimum product, maximum product. Is that okay? To maximize the product, the rate of reaction. So denaturation is denaturation is the disconfiguration of the enzyme into something different that can no longer help to digest or break down the food. Is that okay? Okay, the active site will be lost once an enzyme is denatured. So here are some questions. You can pause the video, like, share, and subscribe, and answer these questions as they come. You do carry out this practical as an investigation, and then here are some investigation you can do on your own, and then here are questions you can practice. And then with this, we've come to the end of our today's class. Thank you for joining us.